Everyone who's tuning in. Good afternoon, everyone who's tuning in. We're happy to see you here on this, what is in the Northeast, a brisk early or late fall, I call it early winter day. I'm Amy Ratner. I'm the Director of Scientific Affairs at Beyond Celiac, and I'm going to be joined by my co-host, Kate Avery.
Hi, everyone. I'm Kate. I'm the Director of Research and Patient Engagement at Beyond Celiac. And today our town hall is sponsored by certified gluten-free Crunchmaster. Crunchmaster crackers are made from simple, wholesome ingredients, and that's about it. What's not? GMOs, gluten, and artificial flavors. Visit crunchmaster.com to learn more and to buy crackers. And they're probably also available at your local grocery store. I know they're at mine.
And from there, we'll move on to introduce our guest who we're quite excited to have join us today and to talk about his work. We're welcoming Dr. Arnold Han from Columbia University. At Columbia, he's an assistant professor of medicine in digestive and liver diseases and microbiology and immunology. So you can imagine that immunology is where we will be going today. Dr. Han was awarded the 2022 Beyond Celiac Early Career Investigator Grant to investigate the role of CD8 T cells in celiac disease. You'll get to learn about those today. And he's going to talk about what he's discovered so far through his research and where he hopes to go in the future. So welcome, Dr. Han. We're excited to hear about the project that you have underway.
It's great to be here, and it's always great to interact with the Beyond Celiac and the celiac community as a whole, which has been just so supportive of my research going back, since my time at Stanford.
So I think probably the best place to start really is to just get an overview of what your study is about and why it's important to people who have celiac disease. Sometimes there can be a little bit of a distance between what's happening in the lab and what people are concerned about in their everyday lives with celiac disease, even as they look to things that might help them in the future. So if you can describe what your work is all about.
Yeah, I think that's a great question. I think that the first step to designing better therapies for any disease is to better understand those disease processes. So that's exactly what, I try to do. And the way I structure my research is to better understand diseases like celiac disease with the end hope of ultimately figuring out ways that we can intervene upon them. And this is what we've done. We've leveraged a lot of unique opportunities afforded to us by celiac disease, namely, it's the only autoimmune disease for which we know what the trigger is. So we have patients at, Columbia and elsewhere within different stages of disease. And we applied these new technologies that have only come, into being in the past 10 or so years to better understand what happens to T cells within different stages of the disease and how they change. And it's our hope that this gives us a lot of clues as to how we might be able to intervene upon celiac disease therapeutically.
So it seemed to me in reading your study that it described the way that gluten triggers two kinds of cells to coordinate and then cause damage. So can you tell us a little bit about how that works?
Yeah, so, we've known for over 20 years now that gluten triggers the CD4 T cell, which is the cell that recognizes gluten, and many of you have probably know about HLA-DQ2 or DQ8, and it's very strongly, that's what people consider to be the celiac gene. And we know that CD4 T cells in patients with celiac disease are the ones that are recognizing gluten in the context of HLA-DQ2 or DQ8. So that's critical to the disease. But what we've grown to understand more recently is that CD4 T cells aren't the cells that ultimately damage the intestine in celiac disease. And instead, they're different types of T cells, namely CD8 T cells and gamma delta T cells. And what we learn, I think, one of the things we learned from our study is that, traditionally it's been thought that CD8 and gamma delta T cells kind of are like bystanders. The CD4 T cells trigger the activation. They're triggered by dietary gluten, trigger inflammation, which in turn activates these gamma delta T cells and CD8 T cells to go and damage the intestine. And reason our study was a little bit surprising is that it kind of showed that gluten somehow directly activates or very rapidly and deliberately activates these other type of T cells as well, CD8s and gamma deltas, the cytotoxic T cells, we'll call them. And that wasn't necessarily well understood before, where I thought, like I mentioned, the previous model was first you get CD4 activation, then you get cytotoxic T cell activation. And our data seems to show that you actually get cytotoxic T cell activation at the same time. And the reason I would say this is a little surprising is that T cells, through their T cell receptors, have the capacity to recognize peptides, like fragments of viruses, for example, or in the case of CD4 T cells, they recognize gluten or gliadin themselves. What's surprising about this is that these cytotoxic T cells, they also have T cell receptors, but nobody in the field actually believes they recognize gluten. So the question is, how is gluten triggering activation of these other T cells that do not themselves recognize gluten? So that's really an important unanswered question that we're actually actively investigating in our lab.
Yeah, I think it is surprising because, from a patient point of view, we think of gluten as being, if I would, the sole actor here. That, in other words, if gluten is the trigger, it's triggering everything. But it sounds like what you're saying is that it's more like a set of dominoes where gluten is the first domino and then it starts to knock down the other reactions at other phases of celiac disease, at other locations and through other cells. Is that a correct way to look at it?
Yeah, I think that's a very accurate characterization. And obviously gluten is the first domino. And I think that one thing that we've tried to understand, because there actually are patients that have these responses, these CD4 T cell responses to gluten, that actually don't get celiac disease. And in mouse models, you can actually generate CD4 responses to gluten without any significant intestinal damage, which is, like I said, caused by cytotoxic T cells. So I think that there's this question about the steps that lead up to cytotoxic T cell activation. And I think that our data, our work provides some clues as to how that might happen.
Mm-hmm.
So we know in celiac disease that the intestinal damage that these cells causes reverses for the most part on a gluten-free diet, but some of the cells in your study don't reverse the way that they act. Can you tell us a little bit more about why that part is important?
Yeah, that's a really good question. That is true that the intestinal mucosa itself, if somebody is gluten-free and has treated celiac disease, pretty much returns to normal. But it leaves a scar, which is not like a scar in the tissue per se, but the immune cells in that tissue remain altered. And it's a really interesting question why that happens. And our data seems to show, and the data of others seems to indicate that those cells are there. Basically, they're kind of there to actually respond very quickly to gluten. Okay, again, there's cytotoxic T cells that were damaging the intestine, and once the gluten is removed, they kind of calm down a little bit, and they're not actually mediating intestinal damage, but they remain in the intestine somehow. So the way I would liken this is, in terms of viral immunity, I think most of us understand the concept of immune memory, meaning like once you've been infected with the virus, your immune system has this unique capacity to remember, and subsequent infections can be fought off by those immune cells. For instance, once you get an infection of your lungs, you'll develop T cells that sit in your lungs, ready to respond to that virus again if it ever sees it again. And what we think is happening is that these cells, that were responding to gluten somehow are sticking around and to respond again to gluten. Now, this immune system obviously is well-meaning. Our immune system, these cells aren't there to cause disease or cause celiac disease. I think they're just being tricked, okay? So they're there actually, those cells, the reason we immune systems have, our immune system has those cells is to respond against viruses or pathogens. And I think that the immune system is somehow being co-opted in celiac disease by gluten. So what's happening is that once you've seen gluten and once you've reacted against gluten, those cells kind of stick around, they're inactive, and they're primed and ready to go again if they see gluten again. So that's what's happening, I think. And I think it speaks to, you know, the immune system kind of remembering gluten as being a threat and ready to respond again.
Okay, that makes sense.
So is that what you mean when you talk about that there's a role reversal in the cells that go from being defenders to attackers? Is that the process you're describing?
Yeah, exactly. I think that, you know, autoimmune disease is a relatively, you know, historically speaking, the role of the biggest threat to our bodies, you know, if you go back hundreds of years, was infection, right? And now that we've had better hygiene and people are living longer, autoimmune disease has become much more prevalent. But the way our immune systems have evolved and what the primary purpose of our immune systems has traditionally been is to fight off infections. And I think that this is what's happening, where our immune systems, which are there to protect us, are kind of perceiving gluten as a threat, even though it's not. It's perceiving gluten inappropriately as a threat. And it's triggering off all these processes, again, that are meant to protect us, but it's triggering them off detrimentally, obviously, in the case of celiac disease.
And I think that the cells that you talk about in your study that were not thought to respond to gluten. So gluten kicks off the process, but these cells in the intestines, yeah, the cytotoxic cells. So they're not directly responding to gluten. And I think you had said that it seems like there are other triggers. Mm-hmm. Do you have any thoughts on what those other triggers might be, or what has your previous work or your ongoing work showing?
Yeah, this is why I think this is a really interesting question, and you got the fundamental question that we're really actively trying to answer, because it kind of goes against the paradigm of immunology, where the cells, you know, T cells are reacting against particular antigens, and those antigens are triggering those T cells off somehow. But here you have cytotoxic T cells that seem to recognize something else besides gluten that are being triggered by gluten. So how is this occurring? And the short answer is we don't know. So we're trying to figure that out. But, you know, and it's not necessarily trivial to figure out what those other things might be, but you could imagine that could be another avenue for therapy. For instance, if we found that they were recognizing a component of the microbiota, or microbes that are in our gut. You could imagine, for instance, that we could somehow potentially treat celiac disease by going after those microbes or whatever is triggering those cells. So this is and also other types of strategies, like for instance, there have been vaccination strategies that have been thought of in celiac disease, and we're really excited about that potential avenue if we figured out what that antigen was. And we're working, you know, with Takeda and other groups and trying to figure out what that might be. So that's a really interesting question that we're trying to answer right now.
So when you say microbes, are you talking about things like, is it bacteria? Is it a virus? I think you talked a little bit about self-antigens, which are described as normal substances that are just in a person's body.
Yeah.
Those are all possibilities?
Absolutely. So those are, I'm just speculating, but yes, those are very good possibilities for what that might be.
So it's, I guess, important to make clear that when we say that there might be another trigger other than gluten, we're not talking about another food that could be involved or another protein that could be involved. It's, we need gluten is still the dietary culprit, let's call it, that starts that domino effect. Then this other factor that might be involved is something that's more internal.
Exactly. So there's something that is somehow causing those T cells, those cytotoxic T cells, because the T cell receptor, or what the T cell recognizes, largely determines what that T cell becomes. So for some other factor is somehow selecting those T cells to be present in the gut, to be ready to go, primed and ready to go when gluten is encountered by the body, by the immune system. And so, yes, we're really trying to figure out what that is. And it could be a really important clue, and that could give us a really good ways potentially to treat the disease.
One of the types of cells that you talk about a lot are T cell clones. Can you just kind of give us a lay explanation of what T cell clones are?
Sure. So T cell, there are two types of cells in your body that express receptors that have the potential for broad recognition. And those are T cells and B cells, okay? So the idea is that, you could pretty much, any substance in the body, any microbe, any protein, your body has the capacity to actually recognize it somehow with the T cell, okay? And how does that happen? Well, each T cell has a receptor, okay? So if it's one receptor, obviously you're going to be very limited in the amount of antigens. I'm going to call them antigens, but we think about them as like fragments of viruses, fragments of bacteria, gluten, for example. Antigens, okay, I'm going to use that term, is what the T cell recognizes. In order to recognize many different antigens, you need many different T cell receptors. Okay, so this is a problem in biology because our genome is only so big and we can only encode for so many different types of T cell receptors. So how does biology do this? Well, basically biology takes the DNA and only in two types of cells, T cells and B cells, and basically chops it up and rearranges it randomly. Okay, so the body produces T cells, and when they come out, when they get developed, they can express millions, literally 10 to the 15th to 10 to the 20th different types of T cell receptors, because it's every T cell has a random rearrangement. Now, to give you an idea of how big that number is, 10 to the 5th, and this is an estimate of how many different types of T cell receptors your body can theoretically generate. Give you an idea how big that number is. I think I read that there are about 10 to the 18th grains of sand in the world. So there's a tremendous amount of diversity in potential T cell receptors, okay? So that's how your body can basically recognize any pathogen or antigen that you throw at it, right? So what is a clone? So a clone is, so the way, once you generate all these different types of T cells, when you find the one that you want, or it sees the right virus, or it sees, in the case of celiac disease, unfortunately, gluten, what happens is that T cells start dividing. So all the cells that are the product of that division will have the same T cell receptor, okay? So that's so our work, we actually use the T cell receptor as a marker of T cell clones, which enables us, so if we see two different T cells with the same T cell receptor, we can reasonably conclude that they came from the same T cell progenitor way back when. So therefore, we can see how T cells change during different states of disease, because we can say, well, in active celiac disease, we see this particular T cell receptor that looks the same as when this patient had inactive celiac disease, right? And we can see how that particular T cell clone changes. And one way we use this T cell clone is that we had patients for which we had biopsies, and they subsequently underwent voluntary gluten challenge. and we track those T cells in the blood. And by virtue of the fact that we can match their T cell receptors from T cells in the gut to T cells in the blood, we were able to track how these T cells changed in response to gluten, because we can match their T cell receptors. So this is what this T cell clone looked like in the gut, and this is what it looks like in the blood once that patient consumes gluten. So I don't know if I clarified it for you or not, but that's what it means by T cell clones. T cells that share the same T cell receptor, is that they are part of the same clone.
I think in your study it said that because you were able to track these T cell clones in the blood, that you were able to see that they become more inflammatory in the blood.
Yeah.
So what does that mean, or what consequences there because of that?
Yeah, so, when in the beginning, I alluded to the fact that you have these cytotoxic T cells and you have CD4 T cells that recognize gluten. And we know that the cytotoxic T cells are damaging the intestine. But the traditional idea has always been, first you got to get the CD4 T cells to be activated. And then over time, you get inflammation building up, and then these cytotoxic T cells go and cause the intestinal damage. And what this gluten challenge experiment enabled us to determine was that these cytotoxic T cells are activated really quickly, and they go from an anti-inflammatory phenotype in the gut to a very pro-inflammatory phenotype in the blood very fast. 6 days.
Is that going into being an attacker?
Yeah, so they go from being essentially a protector, so to speak, to an attacker really quickly, and with the exact same timing as the CD4 T cells. So this is a really, this kind of goes against what the conventional wisdom was, where you needed CD4s to be activated first, and then subsequently the cytotoxic T cells. Somehow these cytotoxic T cells are being activated really, really quickly. Now, this is not to say, and they're not recognizing gluten. Like, nobody's really shown that they recognize gluten. So this is not to say that the CD4 T cell isn't involved in activating them, but it's not downstream of inflammation, right? We think that maybe the CD4 T cells do indeed coordinate to activate these CD8 or these cytotoxic T cells. But the fact that it happened so quickly and so deliberately was I think a novel finding.
One of the questions that I had when I was reading the part about the T cell clones getting found in the blood later with a gluten challenge was, is there any application of that down the road that could kind of lead us to a new biomarker for either diagnosing or managing celiac disease? Yeah,
so absolutely for diagnosing it. It's been, many in the community probably understand that it's a challenge to diagnose patients with celiac disease who are already on a gluten-free diet. So oftentimes patients experience symptoms to gluten, or their mother has celiac disease and they say, I'm going to avoid gluten. And then they go to the doctor and they say, hey, can you diagnose me with celiac disease? And what we can do is we can do a biopsy, which can be normal, and we can do the antibody tests, which can be normal. And then therefore you can't be diagnosed with celiac disease. And traditionally, the only way to do it is make them eat gluten for like 2 weeks, right? Which most celiac patients don't really want to do. So this could enable a short-term gluten challenge, where you could just have a dose of gluten and see. So this could be used potentially as a diagnostic or a biomarker for sure. And I don't know and I know other people are working on other ways to detect the CD4 T cells in the blood. So I think, I'm surprised it hasn't already, but there probably will be a better way to diagnose celiac disease in patients already on a gluten-free diet. Now, it could also, lead to potential therapeutics, I think, because the idea that these cytotoxic T cells are being activated so quickly and deliberately seems to suggest that there's some specific triggering event, right? It's not downstream, non-specifically, they're not just bystanders just getting triggered by inflammation. There's some deliberate process. And if that process is indeed very deliberate and very intentional, so to speak, then maybe we can intervene upon that.
Okay, that makes sense. There's been a couple questions in the chat that are kind of along these lines, and it's a question that I had too. You mentioned in your study that one of the types of cells is similar looking to types of cells that are activated in rheumatoid arthritis and Crohn's disease. So does this tell us anything, one, about like other autoimmune diseases in general, but then two, maybe why people with one autoimmune disease are more susceptible than to other autoimmune diseases?
Yeah, I think both are absolutely correct. So we know that there are genetic susceptibilities that are shared in different autoimmune diseases, including, celiac disease, for instance, with diabetes, and rheumatoid arthritis. So yeah, that's we know that. So that totally makes sense. And I think that broadly, in terms of immunologic mechanisms, I think our study does provide a lot of clues, because like celiac disease, most autoimmune diseases actually, we hear about DQ2 and DQ8 in celiac disease a lot. It's the celiac gene, so to speak, even though that's kind of not really accurate. It's a celiac susceptibility gene. But most autoimmune diseases, like celiac disease, have HLA associations. They're not nearly as strong, but they are mostly associated with HLA class 2. So it's similar and one interpretation of this is maybe for these other autoimmune diseases, there is a trigger like gluten that we don't know yet that somehow is preferentially triggered on these particular HLA types. Okay, so and the fact that we mount these CD4 T cells that look very similar to these other pathogenic autoimmune diseases, yeah, these mechanisms could very easily be shared, and therefore I think what we learn in celiac disease is absolutely relevant to our understanding and potentially treating other autoimmune diseases.
So another thing that's involved in your study, and I don't know if it's directly related to this work or somewhat more separate, but you're using organoids, which to me is such a cool concept. I've been reading the Magic School Bus books with my grandchildren, and we're talking all about all new science and driving the school bus through the body to learn things. Anyway, that's what organoids make me think of, like some kind of, cutting edge science. And just so people in the audience who aren't familiar with an organoid, they're a tiny 3-dimensional mini organ that gets grown in a petri dish. And in celiac disease, it's from the intestinal cells of celiac disease patients. So if you can just briefly tell us what role they're going to play in your understanding of celiac disease and why they're important and what they bring to the research table, I guess.
Yeah, I think that just this in general, the whole concept of organoids in general speaks to how rapidly science is advancing and technology is advancing. So I think that we should all have great hope that science will figure out the answer to celiac disease and in addition, a lot of other diseases. So, we've already applied cutting edge technologies in terms of single cell sequencing and deep sequencing to celiac disease. And so organoids. So what are organoids? Well, first I'll start by saying celiac disease already gives us opportunities that we don't have with any other autoimmune disease, right? Like we understand the trigger. We have patients not only that have active disease, but also treated disease, right? And most autoimmune diseases, you have active disease and you have controls, and that's it. There's nothing in between, but we have patients on a gluten-free diet. We have patients with active disease. We have control patients. We have patients with potential celiac disease, which are patients that mount CD4 responses, but actually don't trigger intestinal damage. And then we have patients who are bravely volunteer to eat gluten. Okay, so there's so many different states. So already it affords us just really invaluable opportunities, but there's always a limit, right? Of manipulation, of experimental manipulation you could do in people, right? And for this, there's a mouse model, which affords us, a little bit more capacity for experimental manipulation. But I would say that, it's still a mouse model. So no matter how good your mouse model is, and the celiac mouse model, I think is quite good. it's still not a human, right? It's not human immunology. Human, albeit, very similar, it's not the same thing. So what organoids enable us to do is, as you mentioned, it enables us to take tissue out of the body and actually study it outside the body. So there we could do experimental manipulations with it. And so this is technology that has come about, in the past 10, 15 years. And, my wife, who I happen to collaborate very, a lot with in the lab, is an expert on intestinal epithelial biology. So this is just a perfect opportunity to take patients with celiac disease where we take biopsies and we can actually grow their intestines in a dish. And, which is not necessarily trivial, because, you cannot just take a cell out of a dish and grow it. You need certain things in that dish to keep the stem cells happy. So we can take intestines out of patients with celiac disease and grow them, propagate them in the dish. And, very interestingly, those organoids actually, a certain type of organoid we use, which is an air-liquid interface organoid, actually retain the T cells in them too. And we could actually add gluten to the culture, and we can actually see T cell activation and intestinal damage. So we're very excited about this. And I think this will enable us to do a lot of things. For instance, test different agents and block different pathways and block different receptors, which obviously we can't do in people. We can basically do our own clinical trials in organoids, right? And see what therapies may or may not work.
Yeah, it sounds exciting and so interesting to me. So we have talked a lot about your research. I think we can all get a very good understanding about why we're so excited to be funding your work and to see where it's going to lead us in terms of potential new biomarkers, which are signals that your body's sending out that would tell someone that you have a disease. And then also for new therapies and, lots of different directions it can go in. But we probably have to get now to the questions from our audience. They sent in quite a few, so we'll start rolling through those. Some of them will apply directly to your work. Some of them are just general things that people want to know and figure you're a good person to ask with your expertise. So I'll kick this off. Well, I think this might have been answered already. Is the role of CD8 T cells in celiac disease mirrored in other diseases? And if so, what conclusions can you draw?
Yeah, absolutely. Yeah, and as you point out, we did kind of allude to this already. Most autoimmune diseases have class 2 associations, which are presumably, or one interpretation is, they activate CD4 T cells. But in most autoimmune diseases, CD4 T cells aren't ultimately the end effector that is mediating the damage. It's usually cytotoxic T cells, like as is the case of in celiac disease. So I think that our work certainly helps to answer this question. How does CD4 activation enable cytotoxicity or tissue damage by cells besides CD4 T cells? And I think our work provides important clues, and I expect absolutely that it could be generalizable to other autoimmune diseases.
I'm going to ask a question that actually came in the chat just a little bit earlier, Amy, and then I'll ask the next question so I don't throw
no worries.
Is what you saw in patients with active celiac disease, one of the, I guess, kind of groups of patients that wasn't in your study was people with refractory celiac disease, either type 1 or type 2. But would you expect to see something similar in people with refractory celiac disease?
Yeah,
I guess I'll end it there because I don't even know what to.
That's a really good question. And we are very interested actually in refractory celiac disease as well. And thankfully, I mean, especially refractory type 2, very thankfully is quite rare. And we don't see a ton of patients with refractory type 2, which is, for all intents and purposes, it's like a very low-grade lymphoma. And we have done actually some limited analysis on refractory type 2. And the cells that are actually expanded in refractory type 2 are very different than the ones we see in active celiac disease. There are cytotoxic cells, T cells, in refractory celiac disease type 2 that do not look like, they look very similar, but they're not derived from the cytotoxic T cells that we see in typical celiac disease. Now, in refractory celiac disease type 1, that's another really interesting question. I don't know. But for the most part, most of the work in refractory type 1 has not picked out many huge differences in the T cell environments compared to active celiac disease. And we do actually have a few refractory type 1 celiac samples that we are getting ready to analyze at some point.
So the next question that we got from people when they registered was about T cells, and could you just talk a little bit about the difference between the T cell reaction when someone has a virus like COVID? This person was asking about COVID, and then in celiac disease, like how those two things are similar and different. And then I kind of have a follow-up after that.
Yeah. So yeah, so I think that there was, there is interesting data about COVID and celiac disease and potentially a link. And I think that there has been a link between epidemiologic links between other intestinal viral infections and celiac disease. So clearly, yeah, there's something there in terms of, in both cases, you're triggering the immune system. And as I alluded to, I think celiac disease, right, is your immune system kind of being tricked into thinking that gluten is like a virus. So yeah, I absolutely think that it's very similar to what an intestinal virus might do, where you are triggering cytotoxic T cells. And I think that, certain aspects of these cytotoxic T cells that are being triggered by celiac disease, including, arising from gamma delta T cells, they're sitting in your gut. And obviously they're not there in, our guts to cause celiac disease. So they're probably there to respond to viruses. So I think that, there absolutely is something even to be understood about antiviral immunity, the better we understand how these cells are being activated.
So is that one of the ways that, we don't know what causes people to get celiac disease? Just, generally 30 to 40% of the population has the right genes for it. Only 1% of the population has celiac disease. Is that maybe one of, does your immune system kind of get tricked by reacting to other things? You kind of alluded to it. You said, there's some evidence that other viruses might kind of trigger celiac disease, but obviously we don't know how it triggers. This person says
that's absolutely possible, because our immune system, this distinction between what's dangerous and what's not is not trivial. In fact, I think we all kind of understand, the immune system being able to recognize what's dangerous and what's not. We all kind of understand that concept, right? Like our immune system, we have to ignore, like foods. We throw all sorts of things into our bodies every day that are not necessarily harmful. Not to mention the fact that there are trillions of bacteria sitting in our guts, right? That in some cases they're beneficial to us, that we don't want to mount immune responses against. So this question about what's dangerous and what's not is hardly trivial, and it's just amazing that our immune systems do as good a job as they do in distinguishing these things. So yeah, I think that it's possible that maybe an infection of some sort or some trigger actually triggers some danger signal that gets co-opted in celiac disease to actually recognize gluten as being dangerous. So yeah, that's a very, very interesting point, and I think that's very plausible.
I think there's been some work that shows that the retrovirus, I think it's the retrovirus, has a connection. And actually, my daughter has celiac disease. And when we think back on how this all started, she did have a virus before all of the symptoms of celiac disease kicked in. So, when you do your experiment on an N of 1, you say, oh yeah, I think that makes sense. That's how it happened for me. So that's my
but the N of 1 turns out to be, there's actually Yeah, absolutely. We can all, it is an N of one, but the studies have borne it out, and there clearly is a link.
Right, right. So another question that we got was, how does the HLA gene type influence the immune function in terms of CD8 cells?
So that's a really good question. There is no HLA association, HLA class 1 association with celiac disease. Okay, so to, actually, without going too deep down the rabbit hole, CD4 T cells recognize HLA class 2, all right? That's what DQ2 and DQ8 are. They're class 2 HLA alleles. They're different types of class 2. So there are many, many different types of HLA genes, and class 2 HLA genes, but DQ2 or DQ8 are the subtypes that are necessary for celiac disease. Now, there's also class 1 HLA genes, which CD8 T cells see. So CD8 T cells see peptides that are on class 1. CD4 T cells see peptides that are in class 2. There was no particular HLA association with CD8 T cells. So, this is not to say that there isn't an antigen, but I think what this is to say is that the critical trigger is undoubtedly gluten, and that's on DQ2 or DQ8. But there is no association, which means, doesn't mean that the antigen is not important, but to me, it implies that the trigger is probably the CD4 T cell. But but I would still say that the antigen for cytotoxic T cells doesn't mean that the antigen for cytotoxic T cells is not indeed important and won't give us clues and potential avenues for treatment.
So another question that came up in the chat is there a certain, do the cytotoxic T cells have to reach a certain level before they start causing damage? Like, is there a threshold to the damage?
That's a really good question. We don't know, actually, but it's actually pretty plausible that for instance, in potential celiac disease, we can see that patients with potential celiac disease, which, like as I mentioned before, they have the antibodies, they'll have TTG antibodies in their blood. They will mount CD4 T cell responses, but there won't be any intestinal damage. What's interesting about it is that we see that those patients with potential celiac disease do, in fact, mount cytotoxic T cell responses. And those T cells look very similar to patients with active celiac disease, those cytotoxic T cells. But they seem to have less of those cells. So that would, that would provide evidence for this threshold idea. But they're not just quantitatively different, they're also qualitatively different. So, it could be a threshold thing, but it could also be other triggers are missing. So it's a really good question. And the short answer is, I don't know. We don't know yet.
So if someone goes from potential celiac disease to actually having celiac disease, does that change? Do those cytotoxic cells become the same? Because you said that they're different. Do they sort of morph into being the same?
Well, yes, exactly. So some aspects of those T cells look very similar, but they have less they express less perforins. So these are the genes, the effector molecules involved in actually killing the tissue. So they express less of those. So the patients that progress from potential celiac disease to active celiac disease, presumably those T cells are going to become more active somehow. So the question is, what is it that differentiates potential? And, it's another really, like I keep alluding to, the opportunities afforded us by celiac disease that don't exist with anything else. It's really interesting to study potential celiac disease versus active celiac disease to figure out, okay, what is the difference here? And can we actually go after those differences and therefore treat celiac disease that way, right? Like turn active celiacs into potential celiacs, for instance.
Or turn potential celiacs into never celiacs.
Yes, exactly.
I feel like we have about 3 lifetimes of research for you. I hope that you have a lot of postdocs in your lab.
We're going to go from early career to established investigator like really fast. So another question that we got is, have they, I'm not sure who they are, discovered if the scenario with CD8 T cells is exactly the same among families of 2 or more members with celiac disease? I guess what they're asking is the disease in family members exactly the same?
Yeah. We haven't done those particular experiments because obviously we don't have very many. We have a huge patient population, but we haven't really looked in families. But I would imagine they would be probably pretty similar. I think that we do see even in unrelated people with celiac disease that are active. They look very similar to one another, and more so than, for instance, if you had a family member that didn't have celiac disease, for instance, there'd be more commonalities among people with active celiac disease than other types of celiac disease. So I would guess that would hold true for families as well.
So Amy, I'm jumping around in the questions for a minute, because I think this one we can kind of relate back. So we know that different people experience celiac disease differently. Some people have only GI symptoms, some people have non-GI symptoms, some people are asymptomatic entirely. So if the immune system response is happening in the same way, how does it lead to such different presentations in different people?
That's a really good question. I'm a practicing gastroenterologist, and it always amazes me when I do endoscopy, where any gastroenterologist would say the same thing. You'll see patients that are like, "Oh, doctor, I have terrible, terrible pain here," and you look in their stomach and it looks totally normal. And other patients who have like these massive ulcers and terrible inflammation in the stomach, and they feel totally fine, and they don't have any symptoms until they start bleeding or something like that. So there is this idea that, so I think that a lot of it has to do with how you sense what's happening in your gut, and there's been a lot of studies on this, not by me but a lot of other people, that there is not a great correlation between what's happening inside our guts and what we feel. And celiac disease actually has another element thrown into it, where I think that a lot of patients feel symptoms from the immune response and not necessarily what's going on in their guts. Perfect example is like when we gluten challenge patients. I'm always, as a doctor, I'm always curious, like, oh, how'd it go? What did you feel? And I'll get a vast different types of responses from, oh, I felt really nauseous, or I felt, kind of a brain fog, to I felt nothing. And it doesn't necessarily correlate to the immune cells that we see in the gut. So, and I think in that aspect, what I think a lot of people are feeling is related to immune response and inflammation. It's almost like a flu-like effect. Like in terms of flu, you're not necessarily feeling, damage to your lungs, so to speak. You're actually feeling your immune system fighting off the infection. So I think there's so many elements of that that are really interesting. But in general, everybody feels symptoms differently from their GI tract, and I think that people have different feelings of what's going on with their immune systems are doing as well.
And I think that's one of the conundrums that we run into, in that people who are asymptomatic, who think they're perfectly fine on the gluten-free diet, they don't really know what's happening in their gut. And then on the other hand, people who feel very sick don't necessarily have that damage. It's something that we've seen in other studies, and I think is also, well, people don't know what's going on in their gut as clearly, but it's reported by the community as well that one thing doesn't always correlate with the other.
Yeah, absolutely. And that's certainly, it's a challenge to us as clinicians as well, because we're obviously trying to help the patients, in terms of their diseases and their absorption and their vitamins, and making sure that, they're healthy from that aspect. But also we're trying to make people feel better, right? And those two things don't always go together necessarily. And it's a challenge as well as a clinician, I see.
Probably in clinical trials too.
Yeah.
How do those things influence clinical trials?
absolutely.
Another question that we got is, once gluten sets off damage to the intestine, is there anything that you can do to stop it? I guess short of taking gluten out of the diet.
Yeah, well, that's what we're all working on, right? We're all working on ways to prevent the disease or treat the disease. But yeah, if gluten is out of the diet, for the most part, most people do quite well, and most people's intestines completely heal. And even though the immune system or the immune cells in the intestine don't always return back to normal, I think that there is evidence that over time, it takes a lot longer. Perhaps those immune cells even start to normalize over time, we think. But, you know, the fact that those immune cells are there, I don't think is necessarily harmful, especially if you stay off that gluten.
Someone in the chat wants to know how they can send you their gut cells, but I think
Yeah.
just so that you don't have cells showing up on your doorstep, can you talk about how people get involved in your research or research in general at Columbia?
Yeah, absolutely. We have a great group of doctors, obviously here, experts who really specialize in celiac disease, and obviously, you know, our center, if you're lucky enough to be close by, we'd love to take care of you. And, you know, just by nature of the way we collect the cells and what we do with them, my lab is located, you know, 5 minutes from the endo suite. So we get very, very fresh samples. And unfortunately, I'd love to collect samples from all the listeners or all the people on this webinar, but, you know, from practical purposes, it's a little difficult. And of course, we do everything with informed consent and all these things. And I don't think that it'd be easy for us to, you know, figure that out logistically through the country. But thank you, absolutely. You know, I'm so appreciative of the patients who are just, you know, who have been so enthusiastic about supporting the work in terms of, you know, volunteering their samples or even volunteering to eat gluten, undergo a gluten challenge for the sake of research. So I really appreciate that.
And I'll have my my colleagues who are answering the chat, some of the other chat questions, drop a link to our patient registry, Go Beyond Celiac, and also our clinical trials page, because you can find, if you're interested in participating in research and clinical trials, we are helping to recruit for some clinical trials right now. So that's another way that doesn't involve having to go to Columbia, or can involve having to go to Columbia, but doesn't only involve having to go to Columbia. So Amy, I'll let you ask the next question.
Sure. So someone wants to know what impact repeated low-level exposure to gluten has on someone with celiac disease over time. I guess in terms of how the cells are reacting.
It's a really good question. I think short answer is we don't know. We don't know the consequences of periodic low-level gluten exposure. I would say that, you know, there are things that we could measure for the most part, right? Like we can measure vitamin levels and, you know, antibody levels to kind of keep tabs on how you're reacting, your body's reacting, and how your intestine. And obviously we can do biopsies and see how healthy your intestines are. So I think for the most part, we don't know, but I wouldn't think that it would have, you know, provided all these other parameters are okay, like the biopsies and various other parameters that we can measure. I wouldn't think that this would have, you know, long-lasting effects. But again, I can't tell you for certain.
And I guess it depends on how you would define low-level gluten as well. Yeah. Like some people might define it as anything under the 20 parts per million standard for gluten-free food. Other people might describe it as anything below, you know, what low level is means different things.
Right, and everybody reacts differently to different levels of gluten, right? Some people are more sensitive than others. So yeah, that comes into play too, I'm sure.
That actually plays in well to this next question, which is what is the smallest quantity of gluten that can trigger an immune response? Would that differ between people?
Yes, I think it does, because, you know, when we do gluten, for instance, gluten challenge has been shown that, you know, the sheer magnitude of things that we could measure, for instance, like the cytotoxic T cell responses, certainly differ. And we've all seen it in the clinic as well, where we'll have patients who take a very long time once they go on a gluten-free diet for their intestinal biopsies to normalize or their antibody levels to normalize. So I think that that differs in different people, and I think it's different for everybody.
Do you think that at some point in time there might be a way to tailor treatment of celiac disease to that level of response?
Yeah, it's our hope
Mhm.
that therapies, you know, we're going to figure out new therapies, and I would expect that, you know, yeah, these therapies should be, you know, could be effective in different types of people with various doses. I mean, I could see a situation where maybe certain patients might be more challenging to treat depending on how robustly they respond. But, you know, I'm hopeful that we'll figure out how to, you know, treat these people as well.
Mm-hmm.
So someone else says that they have a dual diagnosis. They have EOE and they have celiac disease, and they're wondering if something that triggers the immune response in one disease could also trigger the other disease, if that
It's a
makes
really
sense.
interesting question.
Yeah.
I'm not sure there's an association between EOE and celiac disease, not off the top of my head. And from what we know about the mechanisms, there are different types of cells that are involved. Like EOE is more of a Th2 type response, whereas celiac is a Th1 type of response. So there are some differences in mechanisms based on our understanding. For instance, you know, EOE, we see lots of eosinophils, and EOE can also affect, you know, the stomach, or eosinophilic. You can also get eosinophilic gastritis or duodenitis. And in celiac disease, we don't see eosinophils. We mostly see IELs, like these cytotoxic cells in biopsies. So there are a lot of things about the mechanisms that are different, but the answer I would say is, you know, it's absolutely possible because they're both immune processes, and could certainly, one trigger could certainly possibly, you know, flare up the other disease, I would say. Yeah.
Okay. So we're just about, our hour just flew by. And I know we've had an active discussion in our chat, but I guess how we might wrap this up, Dr. Han, is to ask you what you're most excited about. You know, we're about to go into a new year. What are you most excited about in celiac disease research heading into 2024?
Yeah, I think that, you know, I think that when I started science as a student, you know, I think this concept of actually impacting patients with research was something we all aspired to, but was something that wasn't, quite frankly, wasn't very realistic in most cases. And I think that now things have really changed. I think that we've gone to the point where we can actually realistically try to go from the bench to the bedside. So I'm really excited, and I'm really hopeful that it's not going to be long before we have more effective treatments for celiac disease. And that is along the lines, specifically in terms of the research. I'm really excited. We're gearing up to try to figure out what is that antigen for these cytotoxic T cells. And also, I think that with our organoid platform, I'm really excited to be able to test different ideas as to what might differentiate, or like why do we not get disease or damage in potential celiac disease? And I'm hopeful that could actually lead to other therapeutic strategies for celiac disease. So it's generally a very exciting time. Yeah.
And we're all lucky because Dr. Han is only 1 year into his research using his Beyond Celiac SSCD grant. And so next year can bring a whole batch of new developments and steps toward that goal of having new treatments and a cure. So thank you for joining us. I think, your answers to our questions were great, easy to understand for the most part. And we appreciate you taking the time to sit down and talk to us. As I said, people who weren't able to actually attend, who signed up, will get the recording, and so they can find out all the interesting things that you have to say.
All right. Well, thank you all for having me, and thank you for just, again, thanks to the community as a whole, and your interest in this research.
Oh, and the other thing before we sign off that I should remind everyone is that this event is sponsored by certified gluten-free Crunchmaster. Crunchmaster crackers are made from simple, wholesome ingredients, and that's about it. What's not? GMOs, gluten, and artificial flavors. Visit crunchmaster.com. And as Kate recommended, your local grocery store. All right. Thanks, everyone, for joining us. We appreciate our audience as well as our speaker today. And have a great afternoon.
Thank you.